If you were to walk outside right now, right, and just scoop up a single handful of healthy soil, you would be holding more living organisms in your palm than there are human beings on Earth.
Yeah, it's a staggering thought.
Right, and it looks completely still, just inactive dirt.
Yeah.
But the reality is right now, inside that little handful, there is this brutal, invisible turf war taking place.
Oh, absolutely.
It's this microscopic battle for resources that literally dictates our climate, our food supply, and, well, pretty much the health of the entire planet.
Yeah, I mean, the soil is a profoundly active biological engine. We spend so much time, thinking about the plants and animals above the ground because that's what we see. But the foundation of all terrestrial life is constantly shifting and renegotiating its balance of power in the dark.
Welcome to this deep dive. Today, we are opening up the control panel of that underground engine. We have this fascinating stack of sources in front of us, these really comprehensive scientific reviews and meta-analyses. And they're all focused on one of the most critical, yet frequently misunderstood metrics in ecology.
Right, the soil bacteria to fungi ratio.
Exactly. The B to F ratio.
Or sometimes you will see it written as the F to B ratio. The math is just inverted, but the core concept remains the exact same. It is the fundamental metric that tells us who is actually winning that invisible war beneath your feet.
And the mission for our deep dive today is to decode this microbial ratio. We are going to figure out what it means, how it governs the fast and slow energy channels of the Earth, why human activity is accidentally destroying this balance, and how getting it right could be this master key to sustainable agriculture and climate resilience.
Yeah, there's a lot of ground to cover, literally.
Yeah. Okay, let's unpack this. Because to really get a grip on this ratio, we first have to meet the two microbial heavyweights. Like, who are the actual players we're talking about here?
Right. So to really understand these two groups, you have to look at their completely opposing life strategies. I mean, they're playing two very, very different games in the soil. Let's start with the bacteria.
Okay.
You can think of bacteria as the unicellular sprinters of the microscopic world. Ecologically, they are what we call our strategists.
Our strategists, meaning what
It
exactly?
means their entire biological imperative is built around rapid growth and rapid reproduction, just moving as fast as possible.
And I noticed in the research that their diet reflects that speed, right? Like they don't seem to want to work very hard for their food.
No, not at all. They go straight for the easiest calories available. Bacteria thrive on highly digestible labile carbon. So we're talking about simple sugars, amino acids, and something called root exudates.
Which is when the plants basically feed them.
Exactly. When a plant photosynthesizes, it actually pumps a significant portion of those liquid sugars down through its roots and actively leaks them into the soil specifically to feed these microbes.
Wow. And the bacteria just like swarm those roots to gorge on the sugars.
They do. And this ties into a really crucial piece of microbial anatomy, which is their carbon to nitrogen ratio.
Right, the C to N ratio.
Yes. Bacteria have a very low C to N ratio, usually hovering around 3 to 1 or maybe 5 to 1. That means for every 3 or 4 carbon atoms in their tiny bodies, they hold 1 atom of nitrogen.
Okay, so they are incredibly nitrogen dense.
Very dense. And because their bodies demand such a high concentration of nitrogen, they have to burn through these simple resources extremely quickly to satisfy their nutritional needs.
Just to stay alive.
Exactly. This intense rapid-fire metabolism drives what ecologists call the fast energy channel of the soil. They are the engines of rapid nutrient cycling.
Okay, so those are the sprinters, the fast channel. But on the other side of this ratio, we have the fungi. And, well, they seem to operate on an entirely different timeline.
Oh, completely different. The fungi are the multicellular marathon runners. They're case strategists.
So slow and steady.
Right. Instead of just multiplying as single, isolated cells floating in the soil moisture like the bacteria do, fungi actually build physical structures. They grow in these extensive, microscopic, thread-like webs called hyphae.
Which is wild to think about, these little webs.
It is. And these hyphae can stretch across microscopic caverns and soil pores, physically connecting completely different microenvironments. And physiologically, fungi have a much higher carbon to nitrogen ratio, generally between 10 to 1 and 15 to 1.
So they're built out of mostly carbon, meaning they don't have that desperate, constant hunger for nitrogen that the bacteria do.
Exactly. And because of that structural difference, they target completely different food sources. Fungi secrete these complex extracellular enzymes into the soil to break down tough, highly resistant compounds.
like the stuff bacteria can't digest.
Right. They go after lignin and cellulose, the incredibly dense materials that make up wood, tough plant stems, and dead leaves. The bacteria generally just don't have the metabolic tools to touch that stuff.
Wow.
So fungi slowly dismantle those tough compounds, which is why they represent the slow energy channel.
Reading through the sources, this dynamic painted such a clear picture in my head. I couldn't help but think of it in terms of finance.
Oh, really?
Yeah, it feels like bacteria are the day traders of the soil.
Hmm.
They are looking for quick, short-term returns on easy sugars. They multiply fast, cash out, and move on.
I like that.
But fungi act like long-term index fund investors. They are physically building infrastructure with their hyphal webs, slowly compounding tough, long-term assets over a much longer horizon.
What's fascinating here is that your financial analogy actually holds up beautifully when you start looking at the predators that hunt them.
Wait, really?
Yeah, because the physical and physiological differences between your day traders and your index funds dictate the entire food web that sits on top of them. For instance, in a bacterial-dominated soil, a soil full of day traders, you attract bacterial-feeding predators.
Okay.
These are organisms like certain nematodes and single-celled protozoa.
And they're just, what, swimming around, grazing on this massive population of bacteria?
Essentially, yeah. But here is where the cellular math gets really interesting. A nematode might have a carbon-to-nitrogen ratio of 10 to 1, but it is eating thousands of bacteria that have a ratio of 4 to 1.
Oh, I see.
Right, so the nematode is taking in way more nitrogen than its body can physically use. To survive, the nematode has to constantly excrete that excess nitrogen out into the soil as a waste product.
So by hunting the bacteria, these predators are essentially fertilizing the soil.
They are. They are creating a massive immediate burst of nitrogen right next to the plant roots. It is a very fast but very leaky cycle. The nitrogen is available instantly, but if a plant doesn't grab it right away, it just washes away with the rain.
But the fungal web, our index fund investors, they attract a totally different crowd.
Exactly. Fungal-dominated soils support predators like fungivorous microarthropods.
Which are what, like tiny bugs?
Yeah, think microscopic soil mites. And because the fungi have that higher 15 to 1 C to N ratio, their nutrients are bound up much more tightly. When a soil mite grazes on fungal hyphae, the math is different.
The release is slower.
The release of nutrients back into the soil is much slower and much more sustained. You don't get that massive, sudden flush of free nitrogen. You get a steady, reliable drip over months and years.
And this brings us to a massive revelation in these sources regarding carbon. Because fungi are building this physical infrastructure rather than just consuming and multiplying rapidly, their physical presence drastically alters how the Earth handles carbon storage.
Right. Fungi possess a much higher carbon use efficiency, or CUE, than bacteria.
Yeah, I saw that term come up constantly in meta-analyses. What does carbon use efficiency actually look like underground?
Well, when bacteria consume carbon, their fast-paced metabolism causes them to respire a massive fraction of it right back out into the atmosphere as carbon dioxide gas.
They just burn it off.
They just burn it off. Fungi, on the other hand, incorporate much more of the carbon they consume directly into their physical bodies, and fungal bodies are built incredibly tough.
They use that extra carbon for structure.
Exactly. Their cell walls contain highly resilient compounds like chitin, which is the same material found in crab shells and insect exoskeletons, and complex melanins. These materials take a very long time to break down, locking that carbon in the soil long after the fungus itself dies.
And the sources also highlight a specific substance called glomalin that seems to be like a total game changer for carbon storage.
Glomalin is fascinating. It's a sticky glycoprotein exuded by certain mycorrhizal fungi.
Those are the ones that partner with plant roots, right?
Right, the specific types of fungi that form symbiotic partnerships with plants. You can visualize glomalin as a sort of biological soil glue. As the fungal threads push through the soil, they coat the dirt particles in this glue, physically binding the tiny particles together into larger clumps called macroaggregates.
So they are essentially building little earthen vaults.
That is the perfect way to describe it. Inside these macroaggregates, organic carbon is physically walled off from the bacteria that would otherwise eat it and breathe it out as CO2. So fungi aren't just storing carbon in their own bodies.
They're architects.
Yes,
Yeah.
they are actively playing the role of architects, restructuring the physical environment to create microscopic locked carbon vaults.
And it's not just carbon, right? Because that architectural restructuring provides a massive shield against drought, which seems incredibly vital as global temperatures fluctuate.
It is absolutely vital. Fungi possess a distinct mechanical advantage during dry periods. See, bacteria are essentially aquatic organisms at the microscopic scale. They rely entirely on thin, microscopic films of water that coat soil particles just to move around and absorb food.
Okay.
When a severe drought hits, those water films evaporate. The bacteria are suddenly stranded on isolated islands of dirt. They either dry out and die, or they shut down into a deep, inactive dormancy.
But the fungi aren't stranded because they have their physical networks.
Precisely. Their hyphal threads can physically bridge the empty, dry air gaps between soil particles. A fungal network can detect a localized pocket of moisture or nutrients millimeters or even centimeters away.
Which is huge at that scale.
It is a vast distance, and it can draw that water up and transport it through its own internal plumbing, moving resources across dry zones to keep the entire system, including the plants they partner with, alive.
Okay, wait, I have to ask. If bacteria are just burning through resources, leaking nitrogen, and shutting down during droughts, while fungi are doing all this heavy lifting, storing carbon, building soil structure, and transporting water, shouldn't farmers just try to eradicate the bacteria entirely? Like, why don't we just engineer all of our soil to be 100% fungi?
It's a logical question, but if we connect this to the bigger picture, nature demands both channels to function. You do see extremely high fungal dominance in certain ecosystems. Mature, old-growth forests can have F to B ratios up to 100 to 1.
Wow, okay.
And that works perfectly for a forest, which relies on a very slow, highly retentive, closed-loop nutrient cycle. A redwood tree isn't in a rush.
But our food supply is in a rush.
Yeah.
We don't eat old-growth forests.
Exactly. We rely on highly productive agricultural soils to feed the planet. And those systems require the fast bacterial energy channel. Fast-growing annual crops, like wheat, corn, or soybeans, have explosive seasonal growth periods.
They need food right now.
They need massive amounts of nutrients mineralized and made available in a matter of weeks. If you tried to grow a field of corn in 100% fungal soil, the nutrients would be locked up so tightly inside complex organic matter and those fungal vaults that the crop would simply starve to death.
Okay. So the goal isn't bacterial eradication. The goal is the ratio itself. It's about maintaining a functioning balance between the day traders and the index funds.
Right. The crisis we are facing today isn't that bacteria exist, it's that human action has drastically and artificially skewed the balance heavily in their favor.
Which pulls us right into the agricultural battlefield. Because since fungi rely on these delicate, physical, microscopic, hyphal webs to do their job, they're incredibly vulnerable to physical disruption, which, well, seems to be exactly what modern farming is built on.
Unfortunately, yes, industrial agriculture fundamentally wages war on the fungal network, mostly without realizing it. And the most devastating physical weapon in that war is tillage.
The mechanical plowing and turning of the soil.
Exactly.
Here's where it gets really interesting, because I was trying to picture this while reading the reports, and I kept going back to the idea of the soil as a bustling city.
Oh, I like that.
Yeah, so the bacteria are the individual pedestrians walking around the streets, and the fungi are the physical infrastructure. The highways, the suspension bridges, the buried fiber optic cables.
Right.
So when a tractor drags a massive steel plow through a field, it's like taking a fleet of bulldozers and churning that entire city up. The pedestrians, the single-celled bacteria, they mostly survive the tumbling. You mix them up, but they're basically fine. But the infrastructure, the fungal highways, they are completely shredded.
That is a brutally accurate way to put it. And when those bulldozers shred the fungal highways, the soil loses its ability to transport water and bind carbon. The physical biomass of the fungi drops dramatically.
It just ruins the ratio.
And at the same time, plowing injects a massive rush of oxygen into the soil, which artificially favors the fast-breathing bacteria. But the mechanical destruction of the plow is only half the battle. The chemical disruption is just as profound.
Yeah, the sources spent a lot of time on synthetic fertilizers. How does adding nutrients harm the system?
Well, it comes back to the rules of the game. When we pump concentrated synthetic nitrogen into a field, we artificially lower the overall carbon to nitrogen ratio of the entire environment. Fungi naturally hold a competitive advantage in nutrient-poor environments
Because they can forage.
because their physical networks can forage over long distances for scarce resources, and their enzymes can extract nitrogen from complex, difficult matter.
But if you flood the soil with easy liquid nitrogen, you completely erase that advantage.
You change the rules so that the day traders always win. That flood of easy nitrogen causes a massive, unnatural population explosion of bacteria. And because they process nutrients so rapidly, they mineralize nitrogen much faster than the crop's roots can actually absorb it.
Creating that leaky ecosystem you mentioned earlier?
Exactly. The excess nitrogen simply washes away with the next heavy rain, running off into groundwater and rivers, creating dead zones in the ocean. Or it off-gases into the atmosphere as nitrous oxide, which is a potent greenhouse gas.
Man.
By aggressively plowing and over-fertilizing, we have engineered soils that are hyperbacterial, fast-burning, and incredibly leaky.
But there is a massive pushback against this outlined in the research, right? This movement toward regenerative practices.
Yes. Regenerative agriculture is essentially an attempt to shift the ratio back toward the fungi. By transitioning to no-till farming, you remove the bulldozers. You stop the mechanical shearing of the hyphae.
Giving them a chance to rebuild.
Right. And by adding high-carbon organic amendments like crop stubble or wood compost, you provide the complex, tough food sources that fungi specialize in breaking down.
You are giving the index fund investors the specific type of capital they need to start building infrastructure again.
Beautifully said. And beyond just food and shelter, the sources highlight a critical vulnerability called the mycorrhizal gap.
Right, I read about that.
If a farmer grows a single crop, harvests it, and then leaves that field completely bare for the entire winter, they are essentially cutting the power grid to the city.
Because there are no plants feeding them.
Right. Fungi rely on a living exchange with plant roots to survive. A bare dirt fallow literally starves the fungal network.
So the regenerative practice of planting diverse cover crops during the off-season ensures there are living roots in the ground year-round, basically keeping the power on and the fungal highways operational.
It maintains the biological momentum. That is how you rebuild the aggregate stability and the drought resilience that decades of heavy plowing have destroyed.
Okay, so if I'm a land manager, a large-scale farmer, or even just a home gardener listening to this, the obvious next step is to go test my soil. I need to find out my B to F ratio so I know how to fix it. But the sources make it abundantly clear that measuring this ratio is an absolute minefield of scientific caveats. It seems like the deeper you look into the testing, the messier it gets.
Oh, it is arguably one of the most confusing and poorly communicated aspects of modern soil science. The core issue that the meta-analyses reveal is that the specific method you use to test the soil fundamentally changes the number you get.
Yeah, I was getting completely lost in the alphabet soup of the methodologies. The sources list PLFA, qPCR, amplicon sequencing, or gostral assays. Like, how are we supposed to make sense of these different tests?
Think of it this way. Imagine you want to know how many people are currently active in a city. One way to measure would be to scan the city with thermal imaging to count the heat signatures of every living body walking around.
Okay.
That is essentially what PLFA does. PLFA stands for phospholipid fatty acid analysis. It measures the fragile lipid biomarkers in the cell membranes of living organisms, giving you a snapshot of the living biomass at that exact moment.
Okay, so thermal imaging for living microbes. What about qPCR?
qPCR looks at DNA. Measuring with qPCR is like looking at the city's property records and census directory. You are counting the potential genetic presence, but DNA can stick around in the soil long after the organism is dead.
So you're counting ghosts.
Exactly. And amplicon sequencing, another DNA method, doesn't even give you a total count. It just gives you a relative slice of the pie. It tells you the percentages of who is there, but not the overall volume.
Wow. So if a farmer measures their soil with the thermal imaging of PLFA one year and then uses the city directory of qPCR the next year, those numbers can't even talk to each other.
They are completely incompatible. A PLFA ratio of 2 to 1 means something entirely different than a qPCR ratio of 2 to 1. But the confusion doesn't stop at the testing methods. The researchers also warn against something called the high-score fallacy.
The assumption that because fungi are the infrastructure builders, a higher fungal ratio is always a sign of a healthier ecosystem.
It is a very dangerous assumption. Let's look at the example of a severe drought. If you take a soil sample in the middle of a prolonged dry spell, you will almost always see the fungal to bacterial ratio spike dramatically.
Right. And a farmer might look at that lab result and think, amazing, my regenerative practices are working, the fungi are thriving.
But the fungi aren't necessarily thriving at all. The ratio spiked because the bacteria, which remember rely on those thin films of water, are dying off or shutting down en masse.
Ugh.
The denominator of your equation just collapsed. The fungi are merely surviving the stress better than the bacteria. In that context, a high fungal score is actually a flashing red warning light of intense ecosystem stress, not a badge of health.
That makes total sense. And the sources also brought up soil acidity, or pH, as a massive variable that can manipulate the score.
Yes, bacteria generally despise highly acidic environments. So if a field naturally has a low acidic pH, it will inherently be a fungal-dominated soil. But if a farmer applies agricultural lime to neutralize that acidity and make the soil more hospitable for their specific crop, they remove the chemical constraint holding the bacteria back.
So the bacteria just explode in numbers.
You will see a massive bacterial population explosion. The fungal ratio will plummet, looking like a failure on paper, even though the soil is now much more productive and balanced for the crop being grown.
Okay, so if the testing methods are a maze of incomparables, and a high score might just mean your bacteria are dying of thirst, how is anyone actually supposed to use this metric?
This raises an important question, and the consensus from the scientific literature is crystal clear. The bacteria to fungi ratio must never be used as a standalone target. You do not manage an ecosystem just to hit an arbitrary number on a spreadsheet.
It's a lens to look through, not a finish line to cross.
Exactly. It is a trend indicator. The most scientifically defensible way to use this metric is to track a single piece of land over several years using the exact same measurement method every single time. And crucially, you have to pair that lab data with actual physical observations in the field.
You have to look at the dirt in your hands, not just the paper from the lab.
Ask yourself the physical questions. After a heavy rain, is the water infiltrating deeply into the ground, or is it pooling on the surface? When you dig a spade in, does the soil crumble into rich, dark macroaggregates, or is it compacted like concrete? Are your crop yields remaining stable even during a dry spell?
Those are the things that actually matter.
Right. If those physical, real-world metrics are improving over time, and your lab ratio is slowly shifting to show a higher fungal contribution after years of no-till and cover cropping, then you can confidently say your management is rebuilding the slow energy channel. You have to measure the microbes, but you manage the ecosystem.
That brings all of this together perfectly. For everyone listening, the bacteria to fungi ratio is not a magic universal health score that you can blindly chase. It is a profound, intricate lens through which we can observe the hidden, fast, and slow metabolisms of the Earth. Whether it is the rapid-fire, nutrient-burning day trading of the bacteria or the long-term carbon-vaulting architectural investments of the fungi, both strategies are absolutely required to keep our terrestrial ecosystems breathing and functioning.
They are the yin and yang of the soil food web. One provides the spark, the other provides the structure.
The next time you walk through a quiet forest, or drive past a freshly plowed field, or even just water a potted plant sitting on your windowsill, I challenge you to look at the dirt differently. Remember that beneath your shoes is not dead space. It is an incredibly responsive, dynamic, living network that reacts to every drop of rain, every chemical input, and every physical disturbance.
It is constantly shifting, constantly renegotiating its internal economy based on what we do to it from above.
And as we close out today's deep dive, there was one tiny, almost hidden detail in the sources that I have not been able to stop thinking about, and I want to leave you to ponder it. In the final sections regarding the future of soil biology, the researchers briefly touch on bacteriophages and mycoviruses.
The native viruses that specifically hunt and infect soil bacteria and fungi.
There is a whole other level to this underground war. If this microbial ratio is the true biological control panel for carbon sequestration, drought resilience, and global food security, will the future of agriculture eventually move completely beyond mechanical tractors and synthetic chemical fertilizers?
The implications of that technology are staggering to consider.
Imagine a future where, instead of spraying chemicals, we apply biological viral therapies to massive agricultural fields, using highly specific targeted soil viruses to intentionally suppress a certain fast-burning bacterial population, or to actively tune the bacteria to fungi ratio in real time. Imagine programming the soil to save a dying crop, or to aggressively lock away atmospheric carbon just by introducing the right microscopic phage. It forces you to stop thinking of the ground as dirt and start wondering if the Earth's soil is actually a massive planetary immune system just waiting for us to finally learn its programming language.
It radically changes your perspective on what is possible.
It does. And it brings us right back to where we started. That ground beneath your feet, it only looks still,